Nakalai Tesk, the quality for certainty.
Cell culture has several key steps you'll want to get right. In this video, we'll walk you through what to watch out for when preparing, working, and cleaning up. Prepare an appropriate working environment. Use a clean bench that has been properly maintained according to the instruction manual. If necessary, sterilize the workspace by turning on the UV lamp inside the clean bench before use. Turn off the UV lamp before starting work, as its ultraviolet light is harmful to humans and cells. After turning off the UV lamp, run the circulation fan for about 5 minutes in advance. Check the condition of cultured cells before bringing them into the clean bench. Confirm the growth condition and morphology of cultured cells before use. Check the culture medium for any sudden color changes, the presence of foreign particles, or abnormalities in cell shape and growth rate. Since some types of contamination, like mycoplasma, cannot be detected by eye, make sure to perform regular testing. If contamination is found, take appropriate measures. For example, sterilize the materials in an autoclave and discard them safely. Prepare the work area inside the clean bench. Prepare all reagents and instruments required for the operation in advance, so that you don't have to leave the clean bench during work. Wipe the work surface inside the clean bench with alcohol. Wipe instruments with alcohol before bringing them into the clean bench. As with the instruments, make sure to wipe each reagent bottle with alcohol before placing it inside the clean bench. To minimize the risk of contamination, prepare dedicated reagents for each cell line used in culture. Avoid sharing reagents between different cell lines whenever possible. It's also best not to share reagents with other researchers. If sharing is necessary, divide them into smaller portions to reduce the risk of contamination. Use the clean bench for one cell line per session to avoid cross-contamination or labeling errors. Next, let's go over the key points of aseptic technique. Point 1. Always use pre-aliquoted reagents. Preparing your media and solutions in advance helps keep contamination to a minimum. Point 2. Keep the workspace inside the clean bench organized and controlled. Only place the minimum necessary items inside and keep the work area tidy. Try to work in the center area, since airflow near the front may be unstable. Point 3. During operation, always check your hands and pipette tips to make sure you're not touching or contaminating the openings of culture vessels. Point 4. Open reagent bottles and culture containers only when needed. Avoid passing your hands over open bottles or dishes. Before starting, loosen caps slightly to make handling smoother and faster. When placing dish or tube lids on the bench, the direction doesn't matter. However, if liquid is attached to the inside of the lid, be careful. Drops could fall and cause contamination. Point 5. If any medium or reagent is spilled, wipe it up immediately with alcohol. Leaving spills unattended can lead to microbial contamination. Point 6. Do not talk to the operator during aseptic work. Maintaining focus, a clean environment, and good physical condition are essential for safe handling. Finally, let's finish with cleanup. Remove all instruments and reagent bottles from the clean bench. Invisible splashes may cause contamination, so wipe everything with alcohol before reuse. Wipe the inner surface and both sides of the front glass of the clean bench with alcohol. After closing the sash door, keep the circulation fan running for at least 5 minutes before turning it off. If necessary, turn on the UV lamp for sterilization. Since UV light can damage plastic, avoid leaving it on for long periods. About 1 hour is sufficient.
Nakalai Tesk, the quality for certainty.